disclaimer out there and say, no matter what I say about it, I'm not trying to put any morality on top of this lecture at all, although I may say some funny things about alcohol consumption and some stories of my life where I can't remember days. So here's the basic idea, is that ethanol is one alcohol that we can consume and survive. If we consume methanol, we're in serious trouble. If we consume some of the others, I said butanol or we get in trouble pretty fast because there are metabolites that are extremely toxic to our body. Methanol is the worst. Methanol makes formaldehyde. And what does formaldehyde do to cells? You've been studying the anatomy. What does formaldehyde do? It fixes tissue, right? So when we consume methanol, we're in trouble really fast. But ethanol, instead of making formaldehyde, makes acetaldehyde. And that's also a dangerous molecule, but in small amounts, our body can metabolize it and actually turn it into a very valuable energy molecule. So it turns out to be an okay thing as long as our body is able to process it. The problem comes when we make too much acetaldehyde, or if we've lost the capacity to process that acetaldehyde, which is often the case when people have chronic consumption of alcohol at limits that are too high. But as far as fuel goes, ethanol is not bad. 7 kilocalories per gram. I mean, that's a pretty good fuel source. And if you flip it over, people say, well, I want to start eating right and exercising, and watch my calories. One of the first things they recommend is if you're drinking, cut it out. Because that's 7 kilocalories per gram that you just don't need. There's nothing alcohol gives you apart from fun and excitement and stuff like that. There's nothing it gives you that you can't get from a good balanced diet of veggies and fruits and all that kind of stuff. So the cool thing about ethanol is it's a small molecule. It's a CH3, it's a CH2, and it has an OH on it. Real simple molecule right there. And because it's small and with that oxygen on there, it is water soluble. So it'll go in our cells. But it's also just enough nonpolar that it goes right through the cell membrane. It doesn't need a transporter. And so when we consume alcohol and we absorb it, it absorbs readily in our digestive tract. It goes across that cellular membrane. It gets in the blood. It's very happy in the blood. And it permeates all of our cells and permeates all of our neurons and stuff. And that's where the fun comes in. It's kind of fun. Okay? But the liver then is going to be the primary spot where the alcohol is metabolized, the primary spot. And so as you recall from digestion, I think maybe, when we consume a meal, the stuff that's absorbed in the blood goes to the portal vein and it goes up to the liver. And the liver is the first thing that wants to sort of check off and see what's going through the blood. And a lot of times the liver is going to pluck out things that it thinks are necessary to pluck out. And one of those things, of course, is going to be alcohol. So if you have a couple of beers or a cocktail or a keg, whatever, the liver's going to do the best it can to pull that alcohol in and metabolize it, but it's going to take time, which is why they say if you drink a beer, for example, and you sort of feel a little tipsy, it's going to take an hour or so before your body can get back to normal. It just takes time to get that alcohol up and out of your blood. But the process is pretty easy because it just goes right across the cell membrane and right into the liver cell. And we have this enzyme called alcohol dehydrogenase. It's on the next, all the words on the next page you'll see. Alcohol dehydrogenase, and that's out in the cytoplasm. So it gets first crack at that ethanol molecule. And what you see is it takes an NAD, which is oxidized, and it's going to become an NADH because it pulls electrons off of that ethanol molecule. And that's great. You're making NADH, and that's exactly what the body wants. And it's just like the glycolysis, you've made an NADH outside of the cytoplasm, so you've got to get that NADH over to the electron transport chain, but we've got methods for that. Okay, so in the process, when you oxidize an alcohol, what do you make? If you have a single bond OH and you oxidize it, what do you get? A ketone. And that's exactly what happens. The next thing that's made is this acetaldehyde molecule right here. And you see that's a C double bond O right there, acetaldehyde. And because it's an aldehyde, that's why it's so reactive. Now, it's not as reactive as superoxide or hydroxyl radical or anything like that. It is a stable molecule. It does have a finite lifetime. It does have a likelihood of reacting, but under most conditions, if you clean it up in a period of time, it really won't cause much damage. Okay? So we have an enzyme inside the mitochondria here, so the acetaldehyde will be routed into the mitochondria. And we have this saying acetaldehyde dehydrogenase, ALDH. And its job is to pull 2 more electrons off of this molecule, and in the process, make this thing acetate. And all acetate is acetyl-CoA without the CoA, and it's just got that OH on it. So that's the full carboxylic form of the acetate molecule here. And then Wow, love it. I rhythm. And so acetate is a fine molecule. It's not dangerous. It's a carboxylic acid. Of course, it is an acid, but our body has buffers and stuff. And so this molecule is routed out of the mitochondria and out into the blood because acetate is going to be a molecule that cells can pick up. It's kind of like a ketone body, except it's not a ketone. At this point, it's a carboxylic acid. But any cell that has a lot of mitochondria, a lot of mitochondrial power, can bring that acetate in, convert it back to acetyl-CoA, and then use it for energy. So not bad. And of course, a really good customer is going to be our muscles, because our muscles have lots of mitochondria, most muscles, and they'll use that to acetyl-CoA and then do TCA. So it's a really, really nice system. Now, here's the words. And so the two enzymes that we're looking at under most conditions are going to be alcohol dehydrogenase, or ADH. That's a cytoplasmic enzyme. Here's how it works. You're going to oxidize the ethanol to acetaldehyde, and the electrons are going to go to NADH, which is a good thing. And then it's not shown here, but the acetaldehyde is taken into the mitochondria, and it's oxidized again, producing another NADH. This time it's inside the matrix, so that's great. It just runs over to the electron transport chain and gets into electrons. And then here is acetate. Now, a couple of little details here. We have a couple of classes of this alcohol dehydrogenase, class 1, for example, very high affinity. Liver recognizes that alcohol is a molecule that can cause some trouble, so it wants to make sure that it metabolizes it all the way down to nothing. And so if you have a very high affinity enzyme, even when the concentration of ethanol is low inside that cell, the enzyme still has sufficient affinity to bind it and to process it. And of course, acetaldehyde dehydrogenase is going to have the same kind of idea. ALDH2 is going to oxidize 80% of the acetaldehyde. That's inside the mitochondria. And the reason for that is it's not quite as high affinity. It's inside the mitochondria. Things are safer when you're in the matrix and stuff. You have more time to do metabolic processes. But the problem comes in when you have this allylic variant. It's a bad version of this thing where the enzyme is produced and it has the regular fold, but it's not active. And under those conditions, the acetaldehyde is just resident inside the mitochondrial matrix, and it's not being consumed, and so it starts getting all kinds of trouble. And then the other thing that's really good, nature has given us a safety valve. If acetaldehyde is a dangerous compound and it's not being metabolized inside the mitochondrial matrix, it's going to find its way out of the mitochondria and into the cytoplasm. And fortunately, there's a little cleanup enzyme out there, ALDH1, is out in the cytoplasm, and it can pick up any of the excess acetaldehyde. And it will still make the acetate, and the acetate will still go out, and it's okay. And down here is a comment, just like you see before, NADH is going to be very weakly inhibitory towards these molecules. They do produce NADH, but fortunately these molecules are going to be active most of the time, regardless of cellular status, because you want to make sure that you get that ethanol metabolized. And the way our bodies were designed, any alcohol that'll come along. Of course, alcohol dehydrogenase will work on methanol, it'll work on propanol, it'll work on so these are, and we don't want this at all. And so people that have methanol exposure, typically their liver will start to fail very, very rapidly because of this. Just the liver will pick up that methanol and start processing it. And once that happens, there's not much you can do. It's really very damaging. Okay, so this is a little bit confusing, but if we got through it together a little slowly, it'll be okay. The main system for doing alcohol is that alcohol dehydrogenase. So if you like to have a glass of wine with your dinner, if you're having a beer while you're watching the game, or maybe you and your friends are sitting around, you have a couple of beers, no problem. Your blood alcohol level will rise. It will be absorbed by your system. Over a period of time, that alcohol dehydrogenase will chug it, take it down. You have a half a bottle of wine, you're feeling pretty tipsy. You have a 6-pack of beer, you pass out. So what happens then? Okay, so your body says, ethanol is not a molecule that I'm used to. It's a toxin. It's a molecule that comes from outside. It's not a molecule that I make. And so your body also has a detoxification system for molecules that it recognizes as foreign. And believe it or not, lo and behold, it's a cytochrome P450 like we've seen before. So this is in this case, it's called the microsomal ethanol oxidizing system, or MEOS. Let's break that down a little bit. Microsome, when they originally found this, they were taking up cells, and of course they were homogenizing them, and they were looking at the different compartments, and they recognized that some of the small microsomes had the ability to oxidize alcohol. That's where the name came from. But after we got better with cell localization and antibodies and imaging and confocal and stuff, we found out it's in the ER membrane. So the ER membrane is going to contain a cytochrome P450 enzyme has a very high affinity for ethanol, not high affinity, a very high likelihood to bind the ethanol here. And so ethanol will bind to that cytochrome P450. Now, if you remember about a cytochrome P450, what was the basic reaction mechanism? How does the cytochrome P450 work? There were two parts. There was an I'll give you a hint. There was an iron atom, which was where the active site was, and there was another part where some other things were happening. Do you remember any of that stuff? Or you can jump to the next slide and you can shout like you're an expert. Yeah, there it is. So this is the basic idea of the cytochrome P4. We'll go back to the slide. So here's this iron and the heme, and the oxygen atom will bind. If you remember, cytochrome P450s will take an oxygen atom, an oxygen molecule. They'll split those two atoms apart, and one of them will become a I apologize. It binds the oxygen, it turns it into a superoxide. And then that superoxide, one of the oxygens will attack the substrate and bind to it, while the oxygen stays behind and becomes a water. So here is some molecule, any molecule, whatever it is, that binds to the active site. There is the O2, and when it binds to the iron will give an electron to that bound oxygen to create that superoxide. It's hard to see, but that's an O2 minus. And then when the substrate sits down on top of that superoxide, the reaction takes place, and then one of those atoms goes with the substrate. Off it goes ROH. You now put the oxygen on over there, and then the other one is going to just pick up a couple of extra electrons and make water over here. And where do the electrons come from for this process? You have to bring two more electrons in to make that water. What's the source of electrons? Yeah, NADPH. Because NADPH is that synthetic source of electrons, but it's also the source of electrons for our detox system. And the detox system is really amazing. It's how we process drugs. If you get things in there that are poisons, xenobiotics that are in there, the way we process our molecules is through this system here. And some cytochrome P450s are better for, like hydrophobic drugs, and some are better for ethanol, and some are better for, like carcinogens or something. There's a whole different bunch of classes of these things. Okay, so the MEOS system then will say, I'm taking this ethanol, and I'm going to make a superoxide with this oxygen here, and I'm going to use those electrons. Here's the NADPH, so it all comes together, but wait. This molecule already has an oxygen on it. Remember the slide after this? You take a molecule that really doesn't have any oxygens, and you end up splitting that molecular oxygen, and one oxygen ends up on the substrate, and one ends up on water. But this is a little different because this is the one that's made to take care of ethanol or alcohol molecules. In this case, instead of creating an oxygen on the molecule, it just simply says, I'm going to use the electrons. I'm going to steal two electrons from this molecule to make this ketone right here, the acetaldehyde functionality in this case. And I'm going to use the electrons that normally would come from the iron and the electrons that normally would come from the NADPH. And I'm just going to make two waters instead. You don't have to put one of those oxygens on the substrate here. So that's a little bit different. But the product is the same. This detox enzyme says, okay, there's a lot of ethanol floating around. The alcohol dehydrogenase is working really hard. I need to help out a little bit, so I'm just going to do this detox reaction. And it's going to make the same product, acetaldehyde. And then acetaldehyde will find its way into the mitochondrial matrix, and acetaldehyde dehydrogenase will turn it into acetate and all as well, right? Well, maybe. I don't know. Well, the situation for this one is that the cytochrome P450s, if you recall, they are inducible enzymes. What does it mean to be an inducible enzyme? Not just turned on, but they can show up. And so we'll have a constitutive amount of these cytochrome P450s present because they're there to sort of take care of business on a regular basis. But if a person is in the habit of drinking a six-pack every day, and this happens, and they have a lot of consumption of alcohol, and they have a large amount coming in, and their alcohol dehydrogenase enzyme is overwhelmed every day, the cytochrome P450 says, well, you obviously need a lot more help. And the genes get turned on, and more copies of this enzyme are made and put into the membrane to help cycle that excess alcohol, to detoxify the system. And as we know, the likelihood of a superoxide escaping from a cytochrome P450, there's a statistical probability. So the more cytochrome P450s you have, and the more active they are, the more likely that statistical probability will tend towards producing superoxide. And that's the purpose of not this one, but this one here, is to remind you that if oxygen binds and it becomes a superoxide, it's held very tightly by that iron, very tightly. It's only when that substrate comes down and they have that reaction taking place. But when you have a lot of cytochrome P450s and you have a lot of activity, the statistical probability is that once in a while the superoxide is going to get away. And if this happens over my day by day, and week by week, and month by month, and year by year, you start seeing a chronic production of radical, and you get a chronic radical damage. Yes?
Could you explain again how this cytochrome P450 is different from other ones?
Okay, so the question is, how is this cytochrome P450 different than others? Thank you. So for the normal cytochrome P450, you're taking a molecule that's very unreactive. It's usually very non-polar. It doesn't have any polar, and it's usually not very reactive. And so most cytochromes are going to use that oxygen as a way to create a reactive center. It's going to put an oxygen atom on that substrate to give you a site for reactivity, but it's also going to give you a site for more polar, make it more polar. And we'll see this again. We're going to see this coming up when we take the molecule bilirubin, which is the breakdown product of hemoglobin. You put a bunch of oxygens on it, you put the glucuronides on it, out they go. Okay. So since ethanol already has an oxygen on it, let's see, where is that? Here. Sorry, here. Since it already has an oxygen on it, you can't put an oxygen on there, another one. So instead, you just say, I'm going to take your electrons away, and instead of giving you one oxygen, I'm just going to make two waters. So your electrons are going to make one water, and NADPH's electrons are going to make the second water.
But it still makes that superoxide?
It still has the propensity to make superoxide because you still have the basic mechanism of the oxygen binding to that iron, and the superoxide is created temporarily at the active site until it reacts. So if you're a normal, healthy, active person with a good diet and even moderate consumption of alcohol, this really isn't going to be much of a problem. For a person that has a cocktail now and again, or a glass of wine, or a glass of beer, or whatever, your body can handle it just fine. So your MeOS system is not really very active because it doesn't perceive an overload of alcohol. It doesn't see it as a toxic threat, and it lets ADH and ALDH do what they're supposed to do. So for most people, it's okay. Again, I'm not saying it's good or bad to drink or not, but if people choose to, their body can handle it. It's okay. But the problem is, this is a detox system, and so as the level of alcohol rises, for a person who doesn't drink on a regular basis, and they go watch the football game tomorrow, and they're so excited, and they aren't paying attention, next thing you know, 6, 7, 8 beers, right? And their body's not used to that level of alcohol. Their cells are going to be completely infused with that ethanol, and so the cytochrome P450 enzyme is going to be turned on really rapidly to try to detoxify what is considered a non-normal load here. Okay, so as we go back over here, then this will help you again. Here's ethanol. In this case, the oxygen is already present, and so you don't need to put one of these oxygens on it. Instead, you're going to make two water molecules. Two of the electrons come from the ethanol molecule and make the acetaldehyde, and two of the electrons come from NADPH that goes through here. Yes, please.
In the event that it does make superoxide, are we also making the two waters or?
Oh, that's a wonderful question. So the question is, wow, I should go back and revise my test. So the question is, if you make a superoxide, are you still making two waters? Well, the water, for the normal one, one of the oxygens ends up on the substrate, and one of the oxygens ends up in water. So if you make a superoxide and you release that, both of those oxygen atoms go away. They go away as a superoxide anion. So you don't get the oxygen on the substrate, and you don't get the water. And then for the ethanol case, you would not get the two waters, because if your superoxide goes away, then you don't have the individual atoms of oxygen to make that water. That's really good. Okay. And as you recall, what are the enzymes that are going to be hanging around the cytochrome P450 network? Yes, please.
But you still get the water, though, if you have a functioning, like the ROS, cleaning system.
Yes, that's exactly what I was asking at the exact same time.
Oh, okay.
What is the normal mechanism we have surrounding our cytochrome P450 area that's going to be helping to guard against this? What do you expect? Catalase, but before that? Superoxide dismutase, right. So we know that there are cytochrome P450s, and they tend to be grouped in areas of the ER, and so they're going to have a lot of superoxide dismutase hanging out around the ER membrane to scavenge exactly those superoxides. And because they're diffusion-limited enzymes, as soon as those superoxides touch that active site of the superoxide dismutase, they're destroyed immediately. So there's no chance they can hit anything else. H2O2 is produced, catalase hits it immediately, and it's destroyed. And so it's a really nice, effective system. The problem we're about to see with chronic alcohol use is that when acetaldehyde is produced and it can't be metabolized, that acetaldehyde is going to be destroying proteins in the immediate vicinity where it's produced, which just happens to be the antioxidant enzyme, superoxide dismutase and catalase. Yeah. Okay, so here's the good news. If you say, I'm going to go on the alcohol diet, like I told you before, you can get a good bit of energy here. So you get a couple of NADHs formed. When you do the ALDH, that's one. And when you get the acetaldehyde dehydrogenase, that's the second NADH. Sure enough, got lots of ATP. And of course, you get the TCA cycle and you get that GTP coming out. For the MEOS, the microsomal ethanol oxidizing system, well, you use one NADPH because you have to provide electrons to that system to make it work. And you get one NADH produced when that acetaldehyde gets processed. So you get a little bit less energy. But the liver cell says, I am perfectly happy with that because I see that ethanol as a toxic threat, and I'm willing to do anything I need to inactivate that ethanol. And so it puts these cellular resources to work. But that's okay, because the NADH is produced and it makes energy, and then the acetate can be used to make energy. So it's not a bad system overall. Now, what's one thing, if you looked at this thing and you thought, okay, what's being produced, what's being produced, what's being produced? And then you think back, if somebody is doing a lot of alcohol metabolism, what are they producing a lot of? What are a couple things that are being produced here that might be of interest to the cell metabolism pathway? What do you think? What's being produced that would be of interest to the cell? Starts with the letter N. Yeah, NADH is being produced, right? So, what does NADH do to our cellular metabolism? What do you think? Yeah. So the TCA says, wait a minute, that's my job. I'm supposed to make NADH, right? And glycolysis, well, I'm supposed to make NADH. And fatty acids, well, I'm supposed to make NADH. And here ethanol is showing up and it's making all this NADH. So the cell, of course, says two things. First of all, it can say, woo, thank you, I get a little break here. I can relax a little bit because somebody else is doing all the hard work. Or it could be pissed off and say, wait a minute, this is my job and you can't have it. So we have two effects that we're going to talk about. The acute effects. What is an acute effect? Is that real cute, looking, yeah, work it, it's cute, yeah. What's acute? Acute? Yeah, work it, come on. Short term, yeah, immediate short term, right. So if you have a beer or a glass of wine or whatever, martini or margarita, whatever you like, Long Island iced tea, that was my big one. Oh God. Anyway. When I go in McAdoo's, so I was in college in the early 80s here, McAdoo's was just built. That's when the road didn't even go all the way like it does now. It was, we had covered wagons, it was great. We had to tie a horse up front. But McAdoo's, and back then, you could drink beer when you were 18, and you couldn't drink alcohol until you were 21, but I remember my 21st birthday, and we went to McAdoo's, and I was introduced to the Long Island iced tea. And so, when I go in there, I can still sit at the same table that I sat at, and I actually remember where I sat, but I don't remember leaving, but that's okay. Ooh, that was something. So the acute effects are that initially when you process that alcohol, the NADH levels are going to rise, and that's okay. That's okay. There's nothing wrong with NADH. It's a great molecule. And so it does suppress the TCA cycle a little bit. It does suppress fatty acid oxidation a little bit. It does suppress glycolysis a little bit, but that's okay. The electron transport chain says, give me my NADH, give me my FADH2, I don't care how you get it, I want my electrons. So the electron transport chain is happy, the ATP synthase is happy, making that ATP, and everybody's happy, okay? And as the alcohol gets metabolized and it goes away, then the TCA cycle starts to rev back up, and okay? So everything is fine. So just be mindful of the fact. So if a person has a beer or a glass of wine or a cocktail, they have just a little effect. Somebody has a six-pack, they have a big effect, okay? And so then you start to think about, well, what happens if you eat a bag of chips at the same time you drink a six-pack of beer? What's happened to all that glucose that's going into your blood? Is glycolysis going to want to process that?
No.
Is fatty acid oxidation going to want to process that fat that you're consuming? What about a person who is fasting and their liver's working really hard to make glucose to keep the blood glucose level where it needs to be? And you down a couple of beers, that NADH level starts to rise. How is that going to affect the liver's ability to make glucose and export it here? So we'll talk about that. Just wanted to plant that seed. Now the chronic effects here, when you have chronic, and this is day by day, week by week, month by month, year by year. We know that over time, when you suppress fatty acid oxidation and you have these calories you don't know what to do with, the liver is going to make fat. It's going to want to export fat. And it's going to want to put it in the blood. But because of the alcohol and the NADH, you completely screwed up the metabolism of the adipose tissue as well. And so the adipose tissue says, I don't want any more fat. I don't know what to do with this. The NADH is screwing up my pathways. The liver says, well, what am I supposed to do with the fat? It is known, so the fat stays resident, and that leads to that fatty liver here, that hepatic steatosis. Okay, and then alcohol-induced hepatitis. What is hepatitis? Did you talk about that in physiology? It's inflammation of the liver, and it's going to come from damage, any kind of damage that causes tissue. So this, in this case, is going to be the acetaldehyde and those free radicals. So you start damaging your cell membranes, you start damaging your proteins, you start damaging the ability of the liver to do what it needs to do. That gives you that hepatitis. And the damage is healed by a process called cirrhosis, which did you talk about cirrhosis at all in pathology? Anytime you have a damage, like in the blood vessel, and you have damage, you make that fibrin and a clot, and you try to make that tight little cap there to protect that tissue. The same thing happens in your liver. These fibrous proteins come out, they try to heal the damage, but they're fibrous and they're tough. They're not flexible, and they cover areas of the membrane that need to be. So cirrhosis is that hardening and that thickening of the tissues here. And most of it is going to come from this damage that we see here. Okay, does that happen if you drink a six-pack when you're watching a football game? No. Does it happen if you go through a tough time and you drink a little bit more than you used to, but after a couple of weeks you say, I need to get my life back together, and you stop drinking? No. But where this really happens is when you have day to day, week to week, month to month, year to year, and that's the real problem here, the real sad case. Okay, the other pathways that we see here now, the reversible effects. As you can imagine, if a person has a couple of beers, a glass of wine, a cocktail, the NADH level goes up, the normal pathways are going to be inhibited, right? TCA cycle, fatty acid oxidation, glycolysis, gluconeogenesis. But what is that NADH doing during that period of time? Is it just sitting there, or is something happening to it? Yeah, your liver is going to use that NADH. It's going to say this is a good molecule, and it's going to use that alcohol NADH to make its ATP, and that NADH will go away. And so then the TCA cycle turns back on, and the fatty acid oxidation turns back on. So these are called reversible or temporary. So the reversible effects are if you inhibit these pathways, as soon as that excess NADH is used to make energy, then these pathways will come back and resume their normal function. And you assume under reversible cases, a beer, a glass of wine, a cocktail, there's really not going to be any significant damage to your tissues probably. So you're okay, okay? Now this is where it gets a little bit confusing here. If you have a high carbohydrate meal at the same time you drink a lot of alcohol and you make a lot of NADH, you're inhibiting the TCA cycle. You're inhibiting glycolysis. And so the insulin is trying to tell your blood and your cells to bring that glucose out of the blood into the cells, but there's no market for it. All those pathways are down-regulated. And so the glucose stays resident in the blood a little longer than it should, the insulin persists a little longer than it should, and a patient could be hyperglycemic. Is that bad? Nah, not for a couple hours. It's okay. It's not a problem. But it's a problem if it's day by day, week by week, month by month, year by year. Or for, yes?
Will the adipose tissue take up an abnormal amount of glucose in that case?
That's a good question. I would assume yes, because it doesn't have the ALDH and stuff like that, making that excess NADH in the cells. I would assume right. That's a good point. Yeah, I'll make sure, but I think that's right. Okay, good. So if you think about a person who's fasting, and they've got the same problem now, what are the starting materials for gluconeogenesis? What are the starting materials? Lactate, amino acids, alanine, right, and glycerol. How many of those depend on NADH in one form or another to do what they need to do? Two of them, right? Pyruvate lactate, it's going to be dependent on NADH, and glycerol to glycerol. That deals when it gets phosphorylated, and then it gets oxidized to glyceraldehyde. So if your NADH is goofed up, your gluconeogenesis can be goofed up by two of your precursor molecules, but three of them actually. Yeah. So if a person doesn't eat and their liver can't produce glucose, then they're going to be hypoglycemic. And as we see with people who have chronic alcohol use, they tend not to have very good nutritional status. Their liver tends to have to work a little harder. These individuals tend to have more hypoglycemia. It's very unusual for them to be hyperglycemic. But again, that can be reversible. Just within a couple of hours of the last ethanol drink, that NADH can come back to a normal level. These pathways will resume normal activity. The liver can make glucose, or glycolysis can function depending on which side you're on here. Where we get into trouble is when the tissue gets damaged, and that is the real problem. So this looks really complicated, and I know when I show you stuff like this, it's just like, oh crap, okay? But I'm telling you here that the questions about this are really gonna be simple. I don't wanna have these really hugely integrated questions like we just had on the last exam. There are 2 exams left, you have a million exams, you're exhausted, you're pissed off, you're ready to have a little break. And so I wanna ask you some very straightforward questions. So, last year, I think you'll see it, so I said, when a person consumes alcohol, which of the following pathways is most likely inhibited? That was the question that came off of this thing, and it was fatty acids and glycolysis and gluconeogenesis, and it was all the above, which we can't say anymore, right? But you see the kind of question that I'm asking. It's a very straightforward question based on this. But let's go through this together here. Let's just talk it through. So it's always nice. So here's ethanol. A person's had a little bit of ethanol. If it's a small amount, that's fine. We know that so this whole thing is the cell, and of course, here's the mitochondria right in the middle. So now we're out here in the cytoplasm. Ethanol comes in, the alcohol dehydrogenase enzyme is in the cytoplasm, so it metabolizes that ethanol molecule to acetaldehyde, which is fine, and the NADH is produced, and that's a good thing. Now, this toxin here shows that the acetaldehyde is a reactive oxygen intermediate, but it's not super reactive, and if it's made in small amounts, then our body can process it, and we're gonna be okay. And you see that it gets routed now into the mitochondria, where the alcohol, acetaldehyde dehydrogenase takes over, makes another NADH, and there's your acetate that is produced. And that acetate goes out, it's routed out to the blood. So far, so good. Okay, no problem. Now, because of that, here's the NADH that's being produced, and here's the NADH on the cytoplasm that's being produced. And so we start looking at some of the other pathways here. Okay? If you're looking at fatty acid oxidation, for example, fatty acids are being brought into the mitochondrial matrix, and we know that one of the products is NADH, is produced as part of that process. But the NADH levels are going to be a little bit high inside here because of the LDH activity. And so that's going to have a direct effect on the NADH that's produced by this pathway. So fats are not going to be oxidized at the same rate they normally would be. Okay? Pretty good. So now, here we've got some fats hanging out inside the mitochondrial matrix. And more fat is coming in. Here comes fatty acids coming in if you're fasting, for example. And the fat comes in and says, hey, I got a message, I'm supposed to go in the mitochondria. I want to get in there. And the CPT1 says, I'd like to help you out, but they're full. There's no more room. You can't go in. The fat says, well, I was told to go in. No, you can't go in. There's no room. And so the fat starts to accumulate and accumulate. It's like a line outside that bar, and the bouncer won't let you in, okay? And so everybody's getting pissed off and looking at their watch and thinking there might be a better party somewhere. And so they say, hey, this is stupid. Let's go do something else. So the liver says, I'm going to take you and package you together as a triglyceride. And that triglyceride is the way we store excess fat in the liver. And once those triglycerides show up, you're on your way to a VLDL, and you jump on that VLDL bus and off you go to some other great party. That's great. Liver's achieved its goal. That short-term accumulation of fat has been dealt with. Well, if there's a lot of VLDL in the blood already because this has been going on for a while, when it comes time for this bus to leave, it can't leave the liver because the blood says, wait, I've got too much VLDL. I don't know what to do with it. You need to stay there. We can't let you out. The liver says, okay. So there it sits. And the liver says, I don't know what to do with this, and that starts storing the fat. So when you have hyperlipidemia, what does that mean? Too much fat in the blood, which is going to cause these things to stay in the liver, which is going to lead to that fatty liver here, okay? Now, what is allowing you to make those triglycerides? Remember the triglyceride synthesis? We start with glycerol, and we have glycerol, or DHAP in this case. Here's this NADH process right there. So NADH is high because of the alcohol dehydrogenase. DHAP is a normal cellular intermediate. You're going to drive this thing towards glycerol 3-phosphate, or you can phosphorylate glycerol coming in from the fat either way. But now you've got all this stuff that's going to allow you to make the triglycerides. So that's great. You've got the starting material you need to handle those fats to make that VLDL. But at what cost? If you take away your DHAP, what you're doing is you're messing with the ability to do glucose here, make glucose if you're fasting. If you're fasting with fats coming in, you're fasting making glucose going out. In this case, lactate to pyruvate needs to favor pyruvate. Lactate, NADH, makes that pyruvate. Pyruvate goes through that first bypass we saw before. It finds its way, into the mitochondrial matrix and goes through that transformation to OAA and comes out, and then you make the, PepCK and PEP or whatever like that. But now, because of the large amount of NADH out there, you're putting pressure on the pyruvate to go towards lactate. You're taking away that pyruvate that needs to go through that bypass to make the glucose. And you're robbing the DHAP that is made when you bring glycerol in from the adipose. Glycerol phosphorylated, It gets oxidized to DHAP and it's on its way back to glucose. But because of the NADH, you're favoring the glycerol 3-phosphate. So now you've got two things that are working very strongly against you. And of course, amino acids are gonna be the same way, because amino acids get turned into pyruvate, which is gonna get turned into lactate. Yes, please.
I think I missed it. What is the source of fatty acids? Why
Mm.
are fatty acids?
Mhm. This is under the condition if you're fasting, and so the liver's gonna be bringing fat in for its energy, but this fatty acid oxidation is gonna be inhibited. And so the fats come in looking to be burned, but they don't get into the mitochondria, so that's why. Okay, that's why it's there. And so that's why the liver would also be trying to make glucose under these conditions.
So does alcohol not, like, change the insulin or glucagon?
I'm not aware of any effect of alcohol, other than the fact that if you're hypoglycemic or hypoglycemic, that's gonna change the level of hormones that you have. And then you can flip it over, you can say, well, what happens if a person is consuming food, if they're consuming carbohydrate? Well, the same thing happens if a glucose comes in and it's on its way by glycolysis down to pyruvate. As soon as that DHAP is formed, we know in the normal glycolytic pathway, it is isomerized to glyceraldehyde 3-phosphate, which then goes down to pyruvate. But this large DHA pool that's there is gonna be robbed by that NADH, and it's gonna be stolen from that pathway and taken back over here to glycerol, not glyceraldehyde, glycerol 3-phosphate. And that's gonna take away that necessary intermediate you have to allow glycolysis to properly function and give you that ATP. Yes?
Sorry.
That's
just to
good.
clarify, if a patient is fasting and, goes on a binge and, just has a bunch of carbs and stuff in here, are they still metabolically fasting after all those booze and stuff?
Yeah, so the question is the metabolic status. And so anytime you have glucose in the blood that is above that 80 to 100, whatever like that, you're considered an fed insulin state, because that carbohydrate will stimulate the pancreatic beta cells and it will stimulate release of insulin. What you find out is that people that have chronically high glucose have a screwed up insulin metabolism, because after a while the insulin cells become either unresponsive or unable to do what they need to do. Yes, please.
This is really silly, but when you drink, why, if you're not increasing your insulin or your glucagon, why are you so hungry? Like, why do you just want to eat everything?
Why do you want to eat everything? Yeah, that's true. That's more the neuronal effects, sort of what you're happening on. Because while alcohol is a really great fuel, it's not a good filling fuel. Anytime you drink a liquid, you feel full for a while, but not the same if you have protein or fats or something that are slowly digested. Real quick, and we'll go.
Does NADH have any effects on feedback loop on the ADH and ALDH?
On the VLDL and the,
No, the NADH can inhibit ALDH and ADH, like high levels of NADH.
right, okay, so they have a very weak effect as an inhibitor on those enzymes. And I think the reason for that is the liver recognizes it needs to process, and it needs to process those molecules. It's more important to get the ethanol transformed to acetate than it is to say, you've got too much NADH and we need to slow down. I think that the desire to detoxify that molecule is more powerful than you've got too much. That's a great question, thank you. I saw another one. Yes.
Sorry, I'm so confused. Why, if like beer has a lot of carbs in it, are you hypoglycemic?
You're not necessarily hypoglycemic. That's the trouble. You don't necessarily, if a person consumes carbs and they have carbs in their blood, then they're considered fed with insulin. Or if they're not consuming carbs, if they're having like alcohol or with, distilled spirits or something like that, or wine or whatever, which is not carb rich, then they're not going to have a lot of carbs in their system. Which is why beer bellies, right? Versus whiskey, which is thin and wasted, right? So that's part of the reason, the alcohol people, chronic abusers, tend to have a very different body type than like beer abusers. So you can see now that you're stealing that DHAP, and so you're losing the ability to do glycolysis. And so when the glucose comes and knocks on the door and says, hey, insulin tells me I'm supposed to come in here, that bouncer says, sorry, you can't come in. There's way too much glucose 6-phosphate. There's way too much glucose. You need to hang out in the blood. And that's the hyperglycemic case. And it really depends on, some people that are chronic users of alcohols will eat a fairly regular diet, but they still drink. And so they've got a lot of calories and they've got a lot of fuel molecules. And then some people who drink a lot will not consume much food. They just pick and snack or whatever. It just depends on the patient. Everybody's different. Yes? That's a really good question. That's very much a good question. So chronic would be more than the casual use. How's that for a nondescript answer? I would say chronic abuser is somebody that's day by day, week by week, month by month, year by year. That's what I would say. Yeah. I know clinically there's a good definition for that. Okay.
You mentioned that
So anytime the NADH level is high in any of these cases here, whatever enzyme that uses NADH, that substrate pressure will cause the enzyme to go in that direction. That's a really good question. Now this was a little hard to see. We're going to do the purines and pyrimidines next. We're going to do the amino acids, and we'll revisit this and see how this is strongly shifted to uric acid, and uric acid is a primary constituent that causes gout. So a lot of times people with chronic alcohol use and abuse, especially beer, causes a tremendous amount of gout because of the purines that are there. Okay, so it looks like we have a lot of slides. Most of it's just redundant at this point. And so the other problem we have is the alcohol-induced ketoacidosis. When you have a lot of NADH, you have a lot of energy that can lead, if this acetate is being formed, it's possible to make acetoacetate, and that large NADH can lead to the beta-hydroxybutyrate. So the ketone bodies can be produced, and they can go out. And anytime the liver has way too much energy, it makes those ketone bodies. And for normal people that fast, a little bit of ketone bodies is fine. The blood buffer system can handle that. But these are organic acids. And the more that go out, the more you release from protons. And oftentimes the blood pH will start to slip lower and lower and lower. So you can get this ketoacidosis over time. So a lot of times when people come into the hospital that are chronic alcohol users, they have that fruity odor on their breath too, because they have an overabundance of ketone bodies in their blood. And so these are just the words. I've already said how this works. This tells you a little bit about the NADH again there. Again, completely, and this tells you about whether you're doing glycolysis, if you've consumed carbs, or if you haven't consumed carbs, if your liver is making glucose. This is the pathway that goes there. So this sounds highly integrated. It is. It sounds very like, what is chronic? How do I know whether they've had carbs or not? I'm not going to ask you those kinds of questions. I promise you it'll be just a very straightforward. If a person who has consuming alcohol has a very high level of NADH, what will be the effect on their glycolysis pathway? That's what I'll ask. And you know that it's going to shift the pyruvate, lactate thing, or whatever. So, okay. This is what that acidosis, that ketoacidosis and lactic acidosis is. All of these things are weak acids. And so the lactate that our red blood cells make under normal conditions, the lactate that our skeletal muscles make under normal conditions, it's a weak acid, it deprotonates, but our cellular buffer and our blood buffer is more than adequate to take care of those protons and buffer them. But when you have a large amount produced over a long period of time, you exceed the capacity of the buffer, and so the cellular pH will drop and the blood pH will drop. Acidosis. You talked about acid-base, right, in physiology. You talked about that. CO2 in the lungs, bicarbonate in the, okay. So acetaldehyde is bad, and it is bad. And so this is the problem that we have. It's an aldehyde, and aldehydes are reactive. And again, small concentrations of aldehydes are never going to be a problem. It's like we don't die because we have glucose in our blood. Glucose is an aldehyde. It has the ability to reduce things. It's a reducing sugar, but we don't die because our body can handle it on a normal basis. The trouble is when you have a huge amount over a long period of time that stays resident in the tissue, the statistical probability is that acetaldehyde will cause a bit of damage. And if your MEOS gets involved, the MEOS is saying, I'm going to process as much alcohol as fast as I can. And so it makes a boatload of acetaldehyde in an attempt to detoxify that system here. And then when you look at this picture, that sort of gives you an idea of what's happening here. The normal route is ethanol that gets processed to acetaldehyde, that goes into the mitochondria, that gets turned into acetate, and out it goes. That's the normal route. Or acetaldehyde, if it's not processed, sometimes it can slip through and it can get out, but there's other systems that will destroy that for us. So under normal conditions, it's okay. But what this is showing you now is when the MEOS is involved, because the MEOS can greatly increase the amount of acetaldehyde that's produced, then the likelihood of making superoxide is higher because it's a cytochrome P450. The likelihood of making acetaldehyde is very high here. And so you see all that stuff we've talked about before. The radicals are produced, they can damage the lipids in the membrane. That makes the membranes very leaky, so you get fluid movement, you get ion movement, that changes the integrity of the cell, the water balance. You can damage the DNA, you can damage the ribosomes, you can damage the protein machinery, and so the liver's no longer able to make those proteins that it needs to make, like albumin, like ApoC2 and ApoE, and those proteins that go on the HDLs and stuff. So this is a kind of cascade failure that can happen over time. And so the damage that is trying to be fixed is, if the membrane is damaged, You bring those fibrous proteins and you lay them down the membrane to try to seal the damage. The membrane goes from a very fluid, a very dynamic state to a very rigid, very solid state. And you've lost the ability to move things across the membrane. And that's true for your organelles, your plasma membrane, and so forth. And it all comes from these acetaldehyde, these free radicals. And we've been through this before. I'm not really going to ask you so much about that. I'm just interested in acetaldehyde here. You want to spend a minute and just do a couple of questions? Okay, let's do that. It'll be pretty quick. It's easier. Okay, which of the following enzymes is or are most often responsible for ethanol metabolism in the liver following consumption of a small amount or small amounts of alcohol? Now, I was told that I'm not supposed to ask questions like this where I have 1, 2, 1 and 2, 1, 1 and so I'll change the format of this. What do you think the answer might be? Normal mechanism for alcohol is what it's asking. What is it? Yeah, absolutely. C. Alcohol dehydrogenase, oops, and acetaldehyde dehydrogenase. That's just a simple definition question. And that's the type of question I'm going to put on the exam. It's because of the end of the semester, lots of exams and stuff like that, I'm trying to come up with what is the question, what is the answer there. If you don't believe me, I know it's okay. Whoops. Okay, a patient has a deficiency in his or her acetaldehyde dehydrogenase enzyme such that it's completely inactive. Which of the following results would be expected? What is acetaldehyde dehydrogenase? What's going to happen if that enzyme is not active? Right, acetaldehyde will build up because nobody's destroying it, right? So what do you expect? Which one of those answers do you think? You'd expect to see increased acetaldehyde. And so you might argue that this would be true. Acetaldehyde could be exported to the blood. And I realize that too as well. So I fixed that on the exam. But yeah, mostly you expect to see that damage. I'm going to go back and I'm going to send Jean all the ones with all the answer slides, and I'll ask her to post those. Now that we're finished all the lectures, so they'll be available so you can see the answers here. One more. Here's metabolism of ethanol produces NADH, causing cellular levels of NADH to rise. Which of the following pathways would most likely be inhibited by NADH produced by the ethanol? So that's just saying, okay, NADH, who's the fact? Which one? What do you think? Yeah, because all of those are NADH producing pathways. We know the TCA cycle is directly affected by NADH by allosteric regulation. Glycolysis is messed up because the DHAP is stolen by the NADH. Fatty acid oxidation is inhibited because of the NADH production.